Genetics Lesson 2.4 Intro to Mendelian Genetics. So I know before we did a lot of the boring stuff like mitosis and meiosis and DNA, but now is where we actually get to apply it and see it sort of come to life, and I find it really interesting. So we'll start off with a bit of a history from where Mendelian genetics came from. So the basic principles were discovered by Gregor Mendel, and he discovered the basic principles of heredity by breeding garden peas in carefully planned experiments. So essentially what he did is he took these garden peas or these pea plants and he breeded them together to see what the two plants together they would make. So this was essentially his experiment. So many varieties with distinct heritable features or characteristics, such as color, and character variations are called traits. So those are sort of two important words in this unit. Relatively short generation time, and each pea plant has sperm-producing organs or stamens, and then egg-producing organs, carpels. So the mating between the plants was controlled, and the cross-pollination, which is the fertilization between different plants, and we'll learn a lot more about that in our plant unit, but essentially cross-pollination is when one plant fertilizes another plant. They're two different ones, and this can be achieved by dusting one plant with pollen from another. So that's essentially the setup of what Mendel did to get his experiment going. So here is essentially how his experiment took place. So I know this looks really confusing right now, but I promise I'll explain it more. So let's pretend that he took a purple flower and he crossed it with a white flower. He tried to see what the next flower or the next generation flower outcome would be. Now I want you to think to yourself, like, what do you think the flower would be? Do you think it would be purple? Do you think it would be white and purple? Or do you think it would be white? Those are all questions that he had to ask himself to see what it would be in the end. So he determined that the dominant trait, I'll write this out over here on the side so we can remember. So the dominant trait was the trait that was expressed in the first generation. So trait we call it the F1, and that just means first generation. The recessive trait meant that it was not the trait expressed in the first generation, and we'll learn more about what these terms dominant and recessive mean, but essentially dominant meant that it was more prevalent over the recessive trait. So here's his experiment continued. So in a typical experiment, Mendel mated two contrasting true breeding varieties in a process called hybridization. The true breeding plants are the P generation, so the parent generation. That's what he called them, the parent generation, because they obviously started it off. And the hybrid offspring of the P generation are called the F1 generation, so like the first generation that was like the product of the parents. So like my parents were the parent generation, and then I am the F1 generation of them. So the first generation experiment, when Mendel crossed a pure purple flower plant. So he has this pure purple one. It is just pure purple. It has nothing else in it but purple. And a pure white plant, so nothing else but pure white. The resulting F1 generation had all purple flowers. So remember when I asked you before what you thought it might be when we breeded together a purple and a white flower. So it turns out all of them were purple. Now there is a reason for this, and we'll sort of get into that later. Now the second generation experiment, Mendel then allowed the F1 generation to self-pollinate. So remember, the F1 is the product of the parents. So he crossed then the purple and the purple. and this produced a following F2 generation. In the F2 generation, 3 out of every 1, 2, 3, 4 plants were purple. So that's really interesting that even though those plants in this generation up here were all purple, how is it possible that they created a white one? It makes more sense for all of them to be purple, don't you think? And it makes good sense that there are no whites here. So how did this white one create this white one down here? Well, this is where those terms recessive and dominant come in. So I just threw a bunch of different terms at you, and right now they probably mean nothing, but I promise they're going to make a whole lot of sense in a second. So like we said before, we had that purple flower and the white flower, and we crossed them together. They somehow gave us all purple flowers. The only way, and what Mendel determined from that, was that the white flower carried what's known as the recessive trait. So the purple flower is so much more powerful. The stuff inside the purple flower is so much more powerful over the stuff inside the white flower that it actually made all of these flowers purple. But let's not forget that all of these purple flowers still have some white hiding in them. It's just not as visible because the purple is so much stronger. So that's why the overall color is purple. But deep inside its genes and alleles, remember we learned about those, there is a recessive allele of white. So that means that the purple is so much more dominant that it's hiding this recessive allele. However, when we cross all these purple flowers together, we get 3 purple and 1 white. How is it possible that this 1 white flower came from all of these purple flowers? Well, it's because the recessive trait that was hiding in all of these flowers, when it all combined together, it actually gave us this white flower. So there was a possibility, even after all these generations, there was still a possibility of it still being white. Remember that the purple equals dominant, so that's why there will always be more purple flowers. But we still have that recessive trait of it being white. So even though we started off with this purple and white, and all of these were white, tiny pieces of white were hiding in here to make sure that there was still 1 or however many that were white. So hopefully that makes a bit more sense about how it was tracked down and sort of made. Here's a picture of the results of the experiment. I'm not going to go into a ton of detail of this, but you can see how the ratios have started to go down. And he did it a bunch of different times, did a bunch of different things like flowers and pea plants. And he started looking at things that were round and wrinkled and tall and short and stuff like that. So eventually he came to a hypothesis. An organism with 2 identical alleles for a character is said to be homozygous. So something that is the same and the same is homozygous. An organism that has 2 different alleles for a gene is said to be heterozygous. So something that is heterozygous looks something like this. Unlike homozygous, heterozygous are not true breeding. Genotype and phenotype. So I know you've heard me say this a few times, but let's actually start to look at the difference. Because of the different effects of the dominant and recessive alleles, an organism's traits do not always reveal its genetic composition. So therefore, we distinguish between the organism's phenotype, so that's its physical appearance. So phenotype is like the flower is purple, the flower is green, the flower is wrinkled, but its genotype is its genetic makeup. So we say what we learned on the last slide. So we say whether it's homozygous, heterozygous, stuff like that. And an example of the flower color, the pea plants, so the big P, and big P, little p. So big P means dominant because it's two big ones, and then big P, little p means recessive because it's one big and one small, but you don't have to worry about that yet. Have the same phenotype or purple, but different genotypes. So now we can start looking at the law of independent assortment. So what this means is that Mendel identified his second law of inheritance by following two characters at the same time, crossing two true breeding parents using differing two characters, producing dihybrids in the F1 generation, heterozygous for both characters. So what a dihybrid cross is a cross between the F1 dihybrids can determine whether two characters are transmitted to offspring as a package or independently. The law of segregation is essentially Mendel's experiment, and it is the only one of two gene copies present in an organism is distributed to each gamete, or the egg and the sperm cell, that it makes, and the allocation of the gene copies is random. So when an egg and a sperm join in fertilization, they form a whole new organism whose genotype consists of the alleles contained in the gametes. So, genetics after Mendel. So I will talk a ton about this in the next few lessons, but essentially what complete dominance is if I have a white flower who is dominant, and I have a red flower who is recessive. I'll just write dominant up here, and we cross them together, it's going to give us a white flower because the white is dominant. That's just like the regular one that we see. Incomplete dominance, however, is if we have a white flower and a red flower and we cross them together, it's actually going to give us a pink flower. I don't have pink as a color here, so I will just write pink. So when we cross, it sort of gives us a mix. So red and white, sort of if you think of mixing colors together like with paint, it would give you pink. Now codominance is when we have, again, a white flower and a red flower, and we cross them together, and it's going to give us a mix. So we can have like a half white, half red sort of flower. So it's sort of like a mix between the two. And that's sort of what those mean. And that's it for Introduction to Genetics.